The first lesson will be simple diffusion. Simple diffusion is defined as the spontaneous movement of a substance down a concentration gradient from high to low, reaching dynamic equilibrium. Simple diffusion takes place when a molecule is transported across the phospholipid bilayer without the influence of a protein or energy. It is a free process that moves gases such as oxygen or carbon dioxide into the cell. The oxygen, which in this case is the blue pieces of paper, will diffuse across the phospholipid bilayer into the cell until dynamic equilibrium is reached. Dynamic equilibrium is defined as a state of balance between continuous processes, meaning the molecules will be constantly moving at equilibrium. So in this case, we have our oxygen on the outside of the cell, and we have our oxygen inside the cell. Once it's realized that oxygen needs to be inside the cell, it will diffuse across the phospholipid bilayer without protein or energy until the equilibrium is reached. So now there's 5 on this side and 5 on this side. Things to remember about simple diffusion would be the molecule movement from high to low numbers without the assistance of protein or energy. Simple diffusion transports gases. The next part of this lesson will be facilitated diffusion. Facilitated diffusion is defined as the passage of molecules or ions down a concentration gradient across a membrane with the assistance of a specific transmembrane transport protein with no energy requirement. Facilitated diffusion is similar to simple diffusion in the sense that the molecules will be moving from high to low concentration. Facilitated diffusion uses a protein, which you can see right here, to transport across the bilayer. It transports ions in large molecules which cannot fit across the membrane. So in this example, we have calcium, represented by the blue dots, which will be transported through a protein. It will be transported through the protein and into the cell. Calcium is used in the body for the contraction of muscles. So we can move the calcium, it moves through the protein, and it comes out on the side. Through So this one also, facilitated diffusion, also finds equilibrium at the end. So you can see 6 on this side. 1, 2, 3, 4. Yep, 6 on the side and 6 on the side. So things to remember about facilitated diffusion would be the high to low concentration gradient and how the ions move without any energy, but it has a protein. So another part of facilitated diffusion would be it also moves large molecules that cannot diffuse across because it's too big. So the large molecule diffuses into the protein and then to the inside of the cell. Osmosis is defined as the diffusion of water across the semi-permeable membrane. Osmosis is the diffusion of water. In this example, there is an unequal amount of molecule, and the water will move from low to high concentration. So, this is our first osmosis experiment. Each side is going to start with 10 milliliters of water. And then they have the selectively permeable membrane, which is indicated by these dash marks. So on the left side we have 5 molecules, and then this is the water level represented by this teal turquoise line. And then on this, so hold on, 5 molecules. And then on this side there's 15 molecules. On the right side. Water will diffuse across the membrane to distribute the space between the molecules. So if you see there's really no space, it's not equally distributed because there's so many molecules and not a lot of water. While over here there's a lot of space, so the water has the potential to move over here to make more space. So as the water diffuses across the membrane, the amount of water will change on each side accordingly. So I drew 2 so I can show you the cause and the effect. So the water level on the left side will drop, while the right side will increase. Now you can see that this side is more spaced out, showing that the water moved, so the water per molecule concentration, it diluted. So at the conclusion of osmosis, the water on the left side will be 5 milliliters, and then the water on this side, which is the right side, will be 15 milliliters. Oop, we're kind of running off. Rewrite that. 15 milliliters. So to check osmosis, you can do some math. So on this side, on the left side, you have 5 molecules and 5 milliliters of water, which 5 over 5 is 1. And then on this side, you have 15 molecules over 15 milliliters. 15 over 15 is 1. So you can see that the osmosis caused the water to go through the semipermeable membrane to increase the water per molecule. So now they're equally spaced, and it's 1 over 1. The definition of active transport is the movement of a substance across the cell membrane against its concentration gradient, mediated by a specific transport protein requiring energy. Active transport is the movement of molecules from low to high concentration, which is different from facilitated or simple diffusion because that is high to low. So this type of diffusion uses a protein, which is labeled here, similar to facilitated diffusion. And this type of diffusion also uses ATP, which is a source of energy, and it stands for adenosine triphosphate. Active transport uses ATP to make molecules transport from low to high, because naturally high will want to go to low, but in this type of diffusion, low will go to high. So in this example, glucose, which will be represented by our blue, is a source of sugar, and it uses active transport to diffuse into a cell. So glucose starts moving into a cell through the protein, and then ATP will power the glucose to make its way all the way down into the cell. So glucose starts traveling, ATP, and it makes it all the way. So in active transport, especially with glucose, they move from low to high, but see how this one's still here? They don't necessarily all move in. They just kind of have a reserve, so there's a higher concentration on this side. So just to go over active transport again, set it up again. Active transport moves glucose and other sugars across the bilayer using a protein and ATP. protein, ATP. It moves from a low to high concentration, which is different from the other types of diffusion that we talked about. So in this, we're going to have 3 sets of phospholipid bilayers, and we're going to go over the differences in all 3. So in simple diffusion, it transports gases such as oxygen or carbon dioxide from high to low without protein or energy. In facilitated, it transports ions such as calcium into the cell using a protein but no energy. And then in active, it transports glucose or other sugars from low to high, so against the concentration gradient, using a protein and ATP. So as you can see, this one doesn't use energy or protein. This one uses a protein, and these two go from high to low. And then this uses a protein and energy from low to high.